So what do we have today? So we have a set of MCQs. So today's MCQs, the purpose of discussing them is that you guys try to understand the way you're supposed to attempt them and the approach you're supposed to take. That's essentially the purpose. Other than that, we're going to discuss the difficult ones. We're going to go over briefly over the easier ones, and yeah, that's basically what we're going to do. So just gonna, okay, so now we have question number 5. So question number 1 is going to come as well, so don't worry about it. So which type of molecule is the end product of translation? Very easy. Tell me. You guys tell me. Be quick.
Amino acid A.
Okay, Abdullah, what do you think?
Yeah, it's also A. Yeah.
Okay, so you guys are incorrect. So do you remember what translation was? Translation was essentially the mRNA, the ribosome, and the tRNA working together to produce a protein, right?
Yes.
And a protein is a polymer. The amino acid are the beads or the monomers for the protein. So the protein is the polypeptide. Do you guys remember chapter 2? This is also knowledge from chapter 2. Do you know what a polypeptide is? What's the difference between, did you guys take my lecture on chapter 2?
A polypeptide is like the chain of amino acid, right?
Yeah.
No, yeah.
And so what is a chain of amino acids? It's a protein, isn't it?
Yes.
Yeah.
So what's the end product of translation? Isn't it a protein?
Yeah.
Yes.
Amino acids aren't proteins, they're just the monomers. They're like glucose, and when glucose is combined together, it can form cellulose and this and that. But the translation end product is a protein. And so essentially all they've done is put in a bit of chapter 2 where they've confused you between amino acids or polypeptides, and you guys did get confused and opt for amino acids, whilst amino acids are the rare form. They're not the end product, they're the starting product of translation. Do you guys get what I mean?
Yes, teacher. Yes. And
okay. So the diagram shows part of the nitrogen cycle. I assume you guys have done the syllabus, so I'm just gonna discuss this. Which kind of bacteria are involved in steps 1, 2, and 3? Let's see. So he says the step 1 is nitrogen gas in the atmosphere converts into proteins in the plants. So this is something you guys also did in your O-level. So how is nitrogen gas converted into protein? Which of the 3 processes occur? Nitrifying, denitrifying, or nitrogen fixation? Which of these occurs when nitrogen gas is converted into protein in plants?
We studied this, but we totally
you don't
forgot.
recall. Okay. So let me make it easier for you. The process is very simple. We were going from, when we were going here, what we were doing was, what happened? Okay, never mind. Let's go back and let's use this annotated using this. Okay. So if you guys remember the process, I talked about this process earlier as well. So the process was very simple. You had N2 in the atmosphere. Obviously, this is gas that was basically sent into the soil. And then from the soil, it's basically used up in the plants. So in the plants, it's used up for proteins, right? So in the plants, there's a process of absorption, but what's this process that converts into this middle product? So this middle product essentially that we have is a number of steps which are given over here, right? So it says protein in plants, protein in dead plants, and ammonium ions. So essentially what's happening is you have N2 over here, and you have ammonium ion over here, and then you have nitrate ions. Okay, and then in the mid, so what's happening is basically you're going from nitrogen to ammonium. This can be this ammonia, basically which is dissolved in water to give us ammonium, to nitrate, to nitrogen again. This is what's happening. And in this cycle, the part where nitrogen is converted into ammonium is by the help, or it's converted from the air into any other state is by help of the nitrifying bacteria. It's nitrification. When this to form ammonium, okay? When this ammonium is converted into nitrate, when this ammonium is converted, so wait, no, never mind. Okay, so okay, wait, never mind. I did a mistake over here. Okay, so this is a nitrifying. So ammonium, so nitrogen, when it's going into ammonium, what is happening is that the nitrogen is being fixed into the ground or into the plant or wherever it is being fixed. So this is known as, this process is essentially nitrogen fixation. Okay. This process is nitrogen fixation. Okay. So then we have two processes. We have two processes left, nitrifying and denitrifying. So nitrogen has been fixed into the ground by nitrogen fixation. The next process in which the now we have ammonia, which dissolved in water to give us this ammonium ion. Ammonia dissolving in water gave us this. Now we have two processes left, which is denitrifying and nitrifying. So it's very simple. When this ammonium will give us a nitrate, it will be called nitrifying. And when this nitrate will give us a product that cannot be used by the plant or organically, it will be called denitrifying. So all you need to remember is when you get N2 again, it's denitrified, because it cannot be used again. Then the only step left is over here, which is nitrifying. Okay. So all you need to do.
The cycle ends at N2?
It ends at N2 again by denitrifying.
But it can again also get nitrogen fixated, right?
Yeah. So essentially you need to keep N2 in mind. If you keep N2 in mind, everything else becomes easier. For instance, let's go over this again. So I'm telling you, keep N2 in mind, and that's what we're going to do. So essentially we're starting again from N2. When I need to convert N2 into usable product, which is X, a product that I can use, I will nitrogen fixate it. I'll fix it. And then when I have to convert it back into N2, I'm denitrifying something. If you remember this, then all you need to do is remember that within this X, nitrification occurs when the ammonium ions or when the first product gets converted into nitrate. And that's basically it. Okay?
Yes.
Okay, got it.
So let's go about that, all those steps in this diagram right here. Let's clear all my drawings and let's see what this diagram here is telling us. So what this diagram here says is nitrogen gas in the atmosphere is converted into proteins in plants, used in protein in dead plant, dissolves to give us ammonium ions. So this nitrogen being converted into something is, we talked about nitrogen fixation, and that will occur by nitrogen fixing bacteria. So we have two options for that. One occurs in one over here occurs in two places, in B and D. The next one is ammonium B. And so instead of the next one, I'm going to see what's denitrifying nitrogen, giving it back. So denitrifying is happening at the last part, which is number 3. So I'm going to just identify that. Number 3 is over here. So that means B is my correct option. So let's see if 2 is in the right place as well. Ammonium ions are being converted into nitrate ions. And I told you denitrification or denitrifying bacteria. Nitrifying bacteria takes place over here. Okay. So even if you don't remember nitrifying bacteria, you should remember nitrogen fixation, and the opposite where nitrogen gas is released, which is denitrifying bacteria. Okay.
Got it. Yeah.
Okay, let's see the next one. We have question number 3. Question number 3 says, immediately after which stage in mitosis in an animal cell does the cytoplasm start to divide?
D, telophase?
Yes, you're right. Okay, question. I hope this is clear because we did this in the last lecture as well, I remember.
Yes, teacher.
Yeah.
Okay, a peptide consists of 10 amino acids of 4 different kinds. What is the theoretical? Okay, so now you need to understand this. A peptide, which is a protein simply, consists of 10 amino acids. So there are 10 amino acids, but there are of 4 different kinds. So what he's trying to say here basically is that in this confusing statement is that there are 10 amino acids in total. AA is my mnemonic for amino acid. So there are 10, but made up only of 4 different kinds. So 1, 2, 3, 4 of 4 different kinds, A, B, C, D only. Okay. What is the theoretical minimum number of tRNA molecules required to translate the mRNA for this peptide? Okay, so all the question is asking for is I need tRNA number of tRNA that will be used to carry these 4 amino acids for the mRNA. Simply that, right? Translate the mRNA for this peptide simply means, okay, give me the number of tRNA that will be carrying the number of amino acids for this translation process to occur. And I told you, each amino acid needs 1 tRNA. And since in nature we have 20 amino acids, we have 20 tRNAs, not more, not less. So for 4 different amino acids, how many tRNAs do I need minimum?
4, but there are
4.
10.
Yeah, 4. Even though the amino acid number is 10, but there are only 4 different kinds. So even if there are, even if there's 2 tRNAs for one kind of amino acid, for instance, I suppose, even then, when there's when there are only 4 different kinds making up this chain of 10, for instance, I say comes twice or B comes twice to give us these 10 amino acids, but essentially the number of amino acids that are different is 4, which means I will need at least 4 different tRNAs to carry these, right?
Yeah. Yeah.
Make sense, Aruba?
Yes, teacher.
Okay, let's move on. Let's see the next part, next question. Okay, so we've done question number 3. Okay, no, we haven't. So question number, okay, no, we have done question number 3. We have done question number 4. Let's see this. The table describes the walls of 3 blood vessels. Blood vessel 1 is thick layer of elastic fibers and smooth muscle. So 3 blood vessels, and we know when I told you guys when we were I don't know if you guys were there, but when I told you whenever he's saying, oh, yeah, we're talking about blood vessels, there's 2 that we've talked about in detail, arteries and veins, and there's a third one called a capillary. Mostly he likes to talk about arteries and veins. So keep that in mind and look for those in the description. Okay. So he says thick elastic fibers and smooth muscle. Which one do you think this is?
Artery.
Artery, right? Makes sense. Okay. Now the next one is no elastic fibers or smooth muscle. So do you see we're supposed to comprehend it as we go, not at the end, because once we reach the end and we haven't comprehended it, the question looks very tough, even though it isn't. Just like the previous one, just like this. If you did not comprehend this one statement and moved on, then you wouldn't be able to answer it. So comprehend this. No elastic fibers or smooth muscles. Which one could this be?
The capillaries. Yeah.
Yes. Thin
Capillary.
layer of smooth muscle with few elastic fibers, the vein.
Veins.
Which muscles are 1, 2, and 3? Must be very easy. I don't even need to do it for you guys. Okay?
A.
Yeah.
Yes. Lovely. Okay. Next question.
Can
Colchicine is a
you zoom in a little bit because we cannot see?
Can I what?
Zoom in.
Oh, zoom in. Sure. Here you go. Is that okay?
Yes.
I said it earlier, but that's fine. Okay. Colchicine is a chemical that stops chromatids from separating during mitosis. I was just remembering colchicine today. It's a drug we use for gout. So if you guys want to go back and study about gout, now is the time. It's also used in pericarditis. So A-level is helping me revise stuff too. Wow. Okay. So which phase will the cell cycle reach and then stop dividing? So let's comprehend the first line. Colchicine, I don't need to know the name. It's just a chemical that stops chromatids from separating during mitosis. Which phase must this be?
metaphase? No.
Stops chromatase.
Anaphase, yeah.
Anaphase.
Stops it.
Okay, so colchicine is a chemical that stops chromatids from separating during mitosis. That's fine. Anaphase. Which phase will the cell reach and then stop dividing?
The cell will reach up till anaphase and then it won't happen then.
Okay, no, but the point of anaphase is
Yeah.
that division has occurred.
Yeah. So yeah, sorry. The metaphase.
Yeah. So
till metaphase.
So do you get what I mean when I say be able to interpret the question well before you start answering it?
Yes, teacher.
Yes, teacher.
Okay, great. Good brain exercise. Because sometimes I feel like students do cheat because it's always easier to cheat, right? So I want to make sure that you guys did not search this up and
No,
your understanding
we
of the
did
question.
not.
Okay, that's great. Which type of molecule? Okay, we've done this one. Why is it coming up again? Okay, new one. The table shows the percentages of nitrogenous bases in 4 samples of nucleic acids. And we have talked about this, so I'm expecting you guys will help answer this for me. Okay. Which base is adenine? So do you remember the pairing at G and C? And this was at a T or a U. Okay, that makes it very easy. They're supposed to be in the same ratios. Do you remember?
Yes,
Yes.
teacher.
I made you solve a question like this in the lecture as well. So if we have U and T, which are the similar things, just different names, which means that what we're gonna have over here is the fact that T and U will be the same. Okay, so let's see what information do we have. So we have A, B, C, and D, and let's see which ones are which. So he's talking about nucleic acids. And in these nucleic acids, what he's doing is he's talking to us. So we should have a total of 100%, obviously, because I told you these are percentages and not solitary numbers. And then we need to also see what we're talking about. For instance, he says over here, so we have sample 1, 2, 3, and 4. And these samples, so which we need to tell which one is a sample, which one is a sample of adenine. Okay, now you need to understand the question well. These are samples. All of these samples, A, B, C, and D, and uracil, these are bases. A is a base, B is C is D. So this MCQ wants you to identify which of these bases is adenine by using a sample set which is correct. So you don't have to, in previous MCQs, we were choosing what's the correct one and what's the wrong one. In this one, all of these options are correct. What we're doing is we're choosing one of them for adenine. We're assigning adenine to them. So if we're talking about, for instance, now they've given us one hint over here, which is uracil. Let's use this to reach some conclusion. So he says, in all of these, if you can notice, uracil is nil, nil, and nil, right? Except one of these, in which uracil is not nil, it's 25, and the other one is nil, which means when we have uracil and not another one instead of it, which means if these are DNA, this is going to be RNA, which means the other one is
T. Yeah.
T. So C, we know, is thymine, whatever it was, okay? Is that clear?
Yes.
No, teacher, it's not clear for me. Wait, what? Nil?
Okay.
And it's
Nil is zeroing.
so.
Let's go over it again. Let's do it once more. So I said that let's go with the information we have. The information we have says that we have uracil, which is nil in three of these. And we know DNAs do not have uracil, right?
Yes.
And then we have RNAs where we have some bit of, we have uracil and no T, right?
Yeah.
So in one of these samples, we have 25% for uracil, which means this sample is an RNA sample, right?
Yes.
Isn't it? Now, since this one has 25 for uracil, But there's one nil written in its block because it cannot have 5 bases. It can only have 4 bases, right? So which is the
Yes.
fifth base, which is 0? It's definitely the base that is being replaced by uracil, right?
Yes.
And
yes.
which base is replaced by uracil in RNA?
T.
T, which means sample C is T.
Okay.
So all of these are T. Like I told you, all of this table is correct. It did not say right answer or wrong answer. It just says which base is adenine, whether A, B, C, D, or uracil, which 5 of these is adenine. And you have to use this sample box given down here, this experiment box, to decide. So out of this experiment box, we used number 3 because we had one information given, uracil. I used it to find out, okay, nail is coming here, uracil is 25 over here, uracil wasn't present here and here, it's present here, which means what is not present here? Nil. And what can nil be? It can be T, right? Another fact. Now, the second thing, we know that AT or AU combined together and GC combined together. Now, if I know that, let's just use the number 3. Let's stick to this one because it looks easier, because it has helped us identify T, right? So if we have uracil 25, then what should be base pairing and be equal to it?
25.
25A should.
Okay, and then he has confused us as in number 3, he has named all of these 25. Can you see that? So
Yes.
now
Yeah.
we have to look for one of these. So number 3 we cannot use anymore. Now I have to look at 1, 2, and 4 to see where one T a number in T is matching with another number. Whatever number it's matching, whatever one it's matching with. T I know is thymidine. All of these 4 values are for T. Now I need to see in which table they are going to match or be closer together to determine A, simply, right?
So it's B.
B. No. Yeah.
Yeah, it's
Okay.
B.
20.
And that's the correct answer.
Wait, what?
Yeah.
23.
Teacher, but why is A close? Like, why is it not just the same, like 30 and 30 and 24 and 24 and 33 and 33? Why is 32 and 33? Why the
Okay,
difference?
so I told you when we were doing the table in the class as well that these are not arbitrary unit. These are percentages and they are ultra centrifuged, which means there is no perfection in them. Aruba, so how did Abdullah say that it's B? Let me tell you how, because I feel like you did not understand how we reached the answer. Okay, so now we know that the number of A, the percentage of A, like I told you, if I have 50% of A in a molecule, then that 50% is going to bind with T, right? So I'm supposed to have 50% of T. Now in this DNA piece, all I'm having is A's and T's. I don't have anything else. However, in a DNA molecule, for example, in number 3, I have A, B, and C, A, B, D, and uracil, right? I have 25%, for instance, A was 25%, then U binding to it is also 25% because 25 units of A will bind to 25 units of T. Now I have 50% units left. And it's very simple that because G and C need to be in equal ratio, so it will be 25G and 25C, right?
Yes, teacher.
So that makes sense. If that makes sense, then what should also make sense is that if I have in T, if I have 30% of T, then the other 31% of mine should be A, shouldn't it?
Yes, teacher.
Because the equal numbers are pairing, then G and C can be either of these. Okay, if I have 24 of this over here, and then I should have something close by. Now, number 2 is also here to confuse you because these are all in line. These are just to confuse you, just like number 3. Then I'll go to 4 number and see if my conclusion is correct. Again, in 4 number, I see that if this is 33, then the next one close to is 32, which means according to number 1 and number 4, my answer should be B. Because see, these are matching up and these are matching up. They're equal to each other, which means they are base pairing with each other. Even if 2 and 3 don't match, 1 and 4 do. So is this clear?
So can we say that A and D is like G and C? Or
Yeah.
Oh, okay.
Okay. If you understand this, then we'll move on. Otherwise, we can talk about it
Yes,
again.
I understand.
Perfect.
I understood.
What are the conditions in a human cell just before the cell enters prophase? Okay, let's see. Before prophase, what do we have? Before prophase, we have all of the phases have occurred. We have this, we have one DNA here, and its replicated brother over here. So what is the number of chromatids I will have?
No. 4.
Okay, so like I told you that, so let's do it like
4, 4.
science. Let's do it like science. I have 23 and these coming from my mom and my dad. Okay, that's true, right? 23 of these and 23 of these, right?
Yes.
Yeah.
And these are chromosomes. So if 46 chromosomes, I have 46 chromosomes, one of this and one of this, 23 plus 23, right? Then how many chromatids do I have?
92. Yeah.
92. So either B or D. Then the number of DNA molecules are equal to the chromatid molecules. Then I have 92 again, right? Will I
Yeah.
have spindle present in prophase?
No.
No.
Okay. And will the nuclear envelope be
Teacher, I think it's not prophase. It is cytokinesis.
cell before the cell enters prophase? Yes.
Yeah. So then it's cytokinesis.
No, cytokinesis is the last
No.
one. This is
It's
G2.
interface.
Prophase. Yeah. So it is interface. Yeah. Sorry.
Yeah, interface. Correct.
Yeah.
Wonderful, Aruba. Okay. Let's see. Let's see the next question. So the next question says, and so this question was also sent in by Tanya. She wanted to know, she didn't understand it initially before we had the lecture. So let's see what's difficult about it. The diagram shows part of a DNA molecule. Which part is a nucleotide? Quickly, this should be very easy.
A.
Aruba?
Which part is a nucleotide? So
Yeah.
it is B? No.
Nucleotide is
Nucleotide is
Take
like
your
the
time,
whole
take your
base
time.
line.
Even correct answers
Yeah,
are answers
so-
for learning. Doesn't matter. Take your time.
It's
Think of the basic definition. What did the nucleotide consist of?
these are like the structures, purines and pyrimidines, right?
What is the structure of a nucleotide? Purines and pyrimidines, I told you, are nitrogenous bases. What are nucleotides? Okay, I want you to go back and study this. I want you to go back, watch the lecture again, study what a nucleotide is made up of.
Okay, teacher.
Did you read the book?
Yes. Yes, I did.
Okay, go and read the book again and Abdullah will tell you the right answer. And yes, Abdullah, your answer is correct.
Yeah, it's the backbone, phosphate, sugar, and then.
Yeah, look at the phosphate, look at the sugar, and look at the nitrogenous base.
Yeah.
Okay, and which nitrogenous base is this? Is this a pyrimidine or a purine?
It's the smaller one, so it's
A
not a double
pyrimidine.
ring.
So I told you pyrimidine, longer word, smaller base, and
Yeah,
vice
yeah,
versa.
yeah.
Okay.
Yeah.
Okay. A culture of bacteria had all its DNA labeled with the heavy isotope of nitrogen. Okay, the culture was then allowed, and we even did this question, remember? Replication, heavy and light isotopes, heavy and light DNAs.
I think we did.
I don't remember.
We discussed 3 types of replication: dispersion.
Yeah. We did discuss that.
Conservative. Yeah, conservative. And it's exactly the same thing. It's written a little bit tougher because I can't teach you this stuff. Otherwise, if I make my lecture this stuff, you'll never understand it. But the question is obviously tougher, which is why we're discussing questions and not just checking them. Okay. So heavy isotope of nitrogen. Okay, makes sense. Nitrogen is used in nitrogenous bases, right? So in the DNA, which is why he's being able to label the DNA with it. Makes sense?
Yes.
Yes.
Again, understand these lines. They can come up in another question as a direct question, okay? They're not there just for you to look at and admire how wonderfully they write questions, okay? They know they write wonderful questions. Okay, the culture was then allowed to reproduce using nucleotides containing normal ones. Okay, so we had the original culture had heavy isotope, and the other new nucleotides had normal isotope. So the original culture will settle lower when ultracentrifuge because heavy, and the new one will settle higher. The DNA was
The top, yeah.
examined using what? Sorry.
No, yeah, it was at the top. I'm saying the normal one.
Yeah, the normal one will settle at the bottom because it's heavier.
Yeah.
And the new nucleotides were then added, and this tube was let to be replicated. And if I don't let it replicate, I put a separate tube and add the new nucleotides, the new light chain DNAs. Those will settle higher than this lower chain, okay? Because they're light
Yes.
chains. The DNA was examined using a centrifuge after 1 generation and again after 2 generations. Okay, so it was replicated. The diagram shows the position of the DNA band at Z in the centrifuge tube when the DNA was first labeled. So the original heavy one. And which pattern would the DNA be found after the first and after the second cell generations? Very interesting. Let's draw the DNA out and see what result do we get. Okay. Now I don't want you guys to assume the answer over here. Do you guys know why? Why do I not want you to assume the answer over here? Because you'll get it wrong. Yeah, I'm pretty sure you guys will. Okay. So the red one is the original heavy chain and the purple one is the light chain. I'm just gonna label the purple one just so you guys remember. Okay. This was replicated. This chain, okay, never mind. I made a mistake. The original one was just the red chain. Oh, the original one, just the red chain, it opened up in the first replication. So we need to do 2 replications, right?
Yeah, they
right?
are. Yeah.
Okay,
Yeah.
so the first replication, this chain. Okay, at the first generation, where do you think we'll have in the test tube we'll have settlement? If the original settlement was heavy over here, where do you think
half is heavy and half is light?
Are you sure?
Because you can see half the light one and
Yeah.
half the heavy one in the strand.
Okay.
Yeah.
Now next part Aruba will answer. Okay, Aruba?
Okay.
Now I open this one up. I open this one up, right? For the second generation. Okay.
Yeah.
I have to open this one up, so I'll have to open the red one up as well. Won't I?
Yes.
Okay. Now, all of the new nucleotides that I'm adding are the light ones, right?
Yes. Yes.
My original chain was red and it was only one type. I added purple chains to it for replication first. And then I added more, I did not add more purple chains, but I let it replicate again without adding more red. I'm letting the purple one be there. So the purple one's going to get used up. Isn't it?
So with the purple chain, you will not add the red one over here?
No, because the red one is the original one. To one sample, I added I added lots of purple nucleotides, all of these purple nucleotide filled chains, for instance, this test tube, my original test tube, to this little bit of sample. Now the sample is a sample. We cannot add more sample.
So the red thing is the dark Z band?
Yeah.
Okay.
The red thing is the dark Z band, and then the purple one is the new thing. And so in the new thing, when we did the first replication, this is the result we got. This is the result we got. Because obviously we know replication, this strand will, what's happening? Okay, we know in replication, this strand will open up and this strand will open up and the purple ones will quickly bind. Then in second replication, again, both of these strands open up let's do this part again. Let's do this, draw this one again. So in the purple one, now in the second, now I'm doing for the second test tube. In the first test tube, I said both of these are half-half. Aren't they half-half?
Yes.
So which means all will be at Y, right?
Yeah, yeah.
And now in the second generation, now this was the first generation, this is the first. Now in the second one, I'm opening these chains up again for new nucleotides to form with them. So this is the red one from this chain, this is the red one from this chain, right? And this purple chain will also open up, both sides.
Yeah.
Both chains open up for second division, Aruba.
They are get further replicated, right? Within the
And
sample.
yeah,
You're
and
not
now
getting-
on further replication, the new nucleotides that are coming in are just the purples one. We don't have more red ones. We're not adding the sample again. Now look at this. So now how many purple chains do we have? Now let's just do simple math. So we have 1, 2, 3, 4, 5, 6. 6 out of a total 1, 2, 3, 4, 5, 6, 7, 8 are only purple. So aren't these 6 purple?
Yeah, they are the light ones.
They are, and the red ones are only 2 by 8.
Yeah.
So what that also means is, I'm telling you in all ways, sometimes he might ignore this question for a second. Sometimes he might just ask you how many purple chains are there in total, or the percentage of the new purple chains, or the percentage of by weight of it. So you should know that there are 6 individual purple chains now, right, Aruba?
Yes, teacher.
And as for the DNAs, look at this DNA. They are just the light ones, aren't they?
Yeah, they are.
Which means they're going to be settling at X. They're very light. Both chains are light. And the other 2 DNAs are the mixed ones, which will be settling at Y.
Because they're middle, like they're not super light and they're not super heavy.
They're in the middle. They're a mixture of both.
Got it.
So after the second generation, we have half at X and half at Y. Is that correct?
Yes.
So was this a difficult question?
No.
Teacher, how would we know that there would be 2 light, 2 purples and then 2 heavy?
By opening them up. I opened them up for you. I did a first division, I did a second division. As I opened them up, you could now count. You have to draw them yourself and count. You can't just imagine it, which is why I didn't ask you the answer right up front. Okay?
Okay, teacher.
Okay. So this is question, we're back to question number 1, and we're almost done. Let's just finish with this question and we're done with the time as well. So the RNA triplet UAG acts as a stop codon, terminating the synthesis of a polypeptide. The diagram shows a strand of DNA which codes for 4 amino acids. Where would a mutation introduce a thymine nucleotide result in the termination of transcription? So we know termination or stop codon is UAG, right?
Yes.
And we need to see, and how do we see it? We look at, we make if this is the DNA, we just simply make an RNA out of it and see, okay, so for T, what's the one for T? A. Tell me the code, tell me the whole codon.
G.
If this is the DNA and this is the mRNA, A, G,
U, G.
Come on, quickly.
A, G, U, C, A, G, G.
GG. Okay. Now, where do I need to terminate this transcription by a UAG? Where do I put, and he's giving us an option at A, which is over here, and at between 1, 2, 3, 4. Okay, 1, 2, 3, 4. over here, and then so on. So where do I add a
B.
thymine nucleotide, which in mRNA will be an adenine nucleotide, right?
Yeah.
Yeah.
The exact opposite base pairing. Where will I need it to make a UAG? It's very simple, right in front of your eyes.
Yeah, B.
B,
UAG.
exactly.
Yeah.
And that's going to be a stop codon. Okay?
Yeah.
Okay. The diagram shows a diploid cell during mitosis. Which stage of mitosis can be seen? Which phase, guys?
Prophase, right?
Prophase. Absolutely. Perfect. Okay, so we have done this one already. We have done this one already. And we need to do this one, these two. Okay. And then we'd be done with the 20 questions for today. Each of the following events takes place during mitosis. In which order do these events take place? Quickly. I need answers. Quickly.
Four occurs first or 5?
Okay. Chromosomes uncoil. Do chromosomes coil or uncoil in the first phase?
No, that's the last phase.
That's the last one. So what's my answer gonna be?
D.
Yeah, perfect. Do I need to go through all of them, or will you
No,
guys
teacher, no.
go through them individually? If you don't understand one of them, then
Yes.
tell me, okay?
Yes, teacher.
Again, which type of molecule is the end product of translation?
It's amino acid.
No, it's polypeptide.
Just kidding. Yeah, I know. It's funny.
Yeah, you were scaring me. That was not funny. No, but like, okay, good one. No, good session. Did you guys understand it?
Yes, teacher.
Yes, teacher.
Perfect. Then this is amazing. I'll upload it for the others who are celebrating Valentine's and aren't taking classes. So very nice. Okay.
Okay.
So tomorrow we'll do theory questions on this topic and then finally finish it. Okay.
Yeah. Okay.
Okay, teacher.
Are there any questions? Is the topic a little bit
Yeah.
better for you guys?
Yeah.
You guys understand it?
Yes, teacher. Yes.
Yeah.
And please go through the book again. Please know the definitions again, okay? Or at least go through my lecture again.
Teacher, there are a few more questions about that nitrogen one, nitrogen 14 and 15. So can I send you those questions?
Yeah,
So
sure. Send them to me. We'll do them first thing tomorrow. Make sure you send them to me with the answers.
Okay, teacher.
That's what
Okay,
I
no
keep
problem.
telling you guys. We can only do as much in these classes. So make sure you send me the tougher questions instead, because when I randomly make the paper, I don't know which ones you guys find tougher or easier. Okay. So send me the ones you find tougher so we can do those.
Yeah, teacher, I will.
But
send
Nana,
you out
because I feel
tomorrow.
like because you guys are finally understanding biology, you guys have stopped taking interest in it. And that's the recipe to disaster.
Yeah. Probably.
Yeah, we are agreeing, but we're not gonna implement it in our life. Okay, no worries. Just make sure you're serious at least next month. Cover your other subjects for now. But March, a serious month, okay?
Yes,
Okay.
teacher.
Okay, best of luck.
Anaphase.
Allah,
Thank you.
bye-bye.